I appreciate that Dr. Fedoruk has given us an excellent overview to start the afternoon off, and I'm going to continue on in a similar vein, considering actually I did my postdoc with Dr. Fedoruk, so we are very similar in what we do. So our learning objectives are simply to gain an appreciation of how microbial metabolism can influence gut health, to understand how gut microbes can modulate immune function, and to recognize the host of different ways in which we communicate with each other. So as you've just heard, we co-evolved with microorganisms. So they are all around us, we are part of them, they are part of us. And bacterial genes, have you heard, really far outnumber the human genes. So if you look at that tiny little blue dot, that's a human genome, 23,000 genes. If you look at that very large dot, that's a human microbiome genes, 100 million genes in there, far outseeing whatever we have. So really, a lot of our genes, if you go way, way, way back when, a lot of our genes actually came from bacteria. So even in our own genome, we have incorporated bacterial genes within us. So it's not surprising that we have developed this whole host of mechanisms to communicate with them. So how do we do it? Okay, so the evidence now really supports a role for gut microbes communicating, as you heard, with the circulatory system, with the immune system, with the brain, and we're very fortunate to have two world experts in gut-brain axis that we're going to hear from later, Dr. Collins and Dr. Marshall, and as well as obesity, muscular function. So many parts of us are really controlled, I shouldn't say controlled, but modulated by the type of bacteria that we have in our gut. Okay, so the species that we have in our gut change from the stomach, the upper small intestine, as we go down into the colon. Not only the type of bacteria that we have change, but also the abundance changes. So gut motility keeps down the number of organisms in the small intestine, but in the colon, where gut motility is much less, they grow and we multiply, and there's a huge biomass sitting in the colon. Okay, you've heard a little bit about this. I want to go into this a little bit more into detail as to how it actually looks in the gut. So where you have this single layer of epithelial cells that forms a barrier in between these trillions of organisms up here, there's a mucus layer sitting here. Right underneath are all the cells of the immune system. We have T cells, we have B cells, we have macrophages, the dendritic cells that Dr. Fedoruk talked about. Within, we have goblet cells that secrete mucus. We have Paneth cells that secrete the defensins. And as well as we have these M cells. And really, the role of the M cells in the gut is really to monitor the microbial environment. These M cells actually, the microbes come down and they look at them, they identify them, and they present them and show them to the immune system. And this whole system together really is a state of tolerance, that the immune system, which is sitting right underneath the epithelium, tolerates these trillion organisms. Okay? And they have built up a lot of different ways in which this tolerance actually develops. And it's when this tolerance gets disrupted that is when you can end up with diseases, say like inflammatory bowel disease. Okay, so what are the lines of communication? Okay, there's a ton of communication between all of these microbes, between the microbes themselves are talking to each other. They're talking to epithelial cells. The immune cells are talking to each other. The immune cells are talking to the epithelium. And the epithelium is talking to the microbes. Okay? Non-stop talking. So how do they use? What are the signals? How is this actually done? Okay, metabolites, as you heard, this is a key role for metabolites that are produced by metabolism off the gut microbes Short-chain fatty acids, such as acetate, butyrate, propionate, are all used as signals to the epithelium. Things like indole are used as a signal. Peptides, polysaccharides, vitamins, GABA, conjugated linoleic acid, all of these produced by the bacteria used as signals to the epithelium. Coming from the other side, we have things like cytokines, chemokines coming, hormones, peptides, all kinds of things that microbes have learned how to recognize even though they're host-derived. Okay, now metabolism, you heard, so sometimes it's not just who's there, it's what they are doing. So you have all these blokes standing around, all these middle managers standing around, but it's actually this bloke down here who's doing all the work. Okay, so you can look at gut composition and say, okay, well, we've got this many firmicutes and this many Bacteroides, but what are they doing? Okay, what is the metabolism? What is going on? And I'm happy that there's so many people involved in nutrition because diet really has a major role in influencing the production of bioactive compounds. Okay? You have the bacteria in your gut, okay? But they need substrate to work. They need a substrate to work on to produce the bioactive molecules that are used for signaling, that are used for health. If they don't get the proper nutrition, they can't metabolize, they can't do their work, and they can't produce what we need them to produce. So diet has such a key role in maintaining health. And I'll show you some more how that works. So microbes, food, you eat food products, we all know that. And they enter into your gut, and they're going to encounter some bacteria in your upper GI tract. And they're going to do a little digestion happening up here. And they're going to move down, they're going to release some products. And then they're going to come into the colon, the undigested products. And they're going to hit this huge biomass that's sitting in the colon. And this biomass is going to metabolize whatever is coming into the colon and produce things like short-chain fatty acids. Okay? If they don't get the proper material coming in, they can't produce these bioactive compounds. And you need kind of an integrated microbial metabolism for health. And what do I mean by that? What do I mean is that you have this microbe A, he's metabolizing something you just ate. He releases something that microbe B uses, passes on to microbe C, and you produce all of these metabolites that you need for health. But you need microbe A, B, and C to produce the molecule. Plus, you need the substrate in your diet to produce it. So if you heard we lost some bacteria in IBD. So what does that happen to some of the metabolism? Okay, you don't have microbe A, so you don't produce what microbe B needs, so you end up producing some other toxic metabolite, or you don't produce one of the beneficial metabolites. And that's how you can end up with a type of disease, because you're missing certain key microbes. And to give you an example, butyrate is a bioactive molecule that's been shown to have very good beneficial effects for us. But it requires cooperation in the gut to produce butyrate. So you need first the production of lactate and acetate, which is then used by certain microbes to produce butyrate. Okay? And the type of microbes that work together are, say, things like the lactobacillus and bifidobacterium, which can produce the lactate, and things like these type of bacteria, which then act on the lactate to produce butyrate. Okay? If you're missing these groups of bacteria, you cannot produce butyrate. And then the butyrate then is transported into the body, and it has a trophic effect, so it's used as energy for colonocytes, maintains barrier function because it's used for energy, It has a regulatory aspect because it can inhibit histone deacetylation, so it modulates cell differentiation apoptosis, so it can have a role in cancer. And then it also has an anti-inflammatory role through its actions on a nuclear factor kappa B. So all of these are beneficial roles for butyrate, but you need the cooperation between all of these different microbes to produce it. Okay, here's another example, tryptophan in the diet. It can be metabolized to produce indole and skatole. And then what happens is that this indole can either maintain gut barrier, or depending on which microbes are there, it can be metabolized to produce several toxic metabolites. So again, if you in your gut have some of those bacteria that can produce these toxic metabolites, that's the way it's going to go. Where if you can replace them with the ones that don't, then you're not going to be producing these toxic metabolites. Here's another example. A Bifidobacterium longum, this was an animal study, where they showed that if they infected mice with E. coli 0157, all the mice would die. Okay? So by 7 days you infect the mice, all the mice died. But if you added on a Bifidobacterium longum that produced acetate, you got 100% success or survival. Whereas if you used the same Bifidobacterium longum that did not have the enzyme to make acetate, what would happen is the mice would still die. So you required this particular strain of bacteria that had the enzyme that they could produce acetate to be protective. So that's kind of a little bit about the metabolism and metabolites that are really beneficial to us. Okay, so what about the immune system? We heard that microbes interact with the epithelium in a variety of different ways. These M cells that I talked about really monitor and see what's going on. These dendritic cells, you heard, stick out their dendrites, pick up and bring back in, as well as directly with epithelial cells. Microbes all have a barcode that the body can read. So depending on the barcode that it reads, you get a response A, B, or C. It's a structural barcode that the body can read and know what it is. And it reads what is in the bacteria, the structural components. So gram-positive bacteria have certain proteins in their structural membranes. Gram-negatives have got different kinds of structural proteins. And the body has developed a way to recognize the outside of bacteria. And again, whatever the bacteria has on the cell surface, the epithelium can look at, recognize, and respond through actions between the immune cells and the epithelium. And it does it through something called toll-like receptors. So there's about 12 different toll-like receptors. What these things are is, this is your cell membrane. It's a receptor. So part of the receptor is inside the cell, part of the receptor is outside the cell. Gram-negative bacteria have something called lipopolysaccharide, flagellin, and there's separate receptors, it's called TLR4, TLR5, that recognize LPS and flagella. And they can know it's a gram-negative bacteria. And same with gram-positive and yeast. We have all of these different toll-like receptors that can recognize it. And same with double-stranded RNA, so they can recognize viruses and they can recognize bacteria that get inside the cell. So these are receptors that read the cell surface of the organism. And again, the barcode. So whatever receptor is activated, you get a different cytokine and chemokine response inside the body. And depending on the strain, microbes can stimulate an immune function, like stimulating IgA secretion or stimulating phagocytosis, or they can induce an anti-inflammatory effect, or they can induce no effect at all. So again, it's very individualized what that particular microbe will induce the epithelium to do. And here's this dendritic cell again. Really controls a lot of things. So when they bring in a bacteria, they read it, and let's say they bring in a Bifidobacterium longum, this is just an example. They will release cytokines like an anti-inflammatory, a TGF-beta or a TNF. Whereas if they bring in a Salmonella, you can see that they release things like IL-10, IL-12, TNF, interferon. So they will induce an inflammatory response against a Salmonella, but not the Bifidobacterium. So very individualized responses. And the way it works is that you have the whole microbiome as a whole. We talked about the balance. So that microbiome, it's got all inflammatory signals in it, it's got anti-inflammatory signals, it's got metabolic signals in it. So what you end up getting is it can drive it to a pro-inflammatory type response in the epithelium, can drive it towards an anti-inflammatory response, depending on which type of bacteria is there over the other type. So if you have more anti-inflammatory, you'll drive it towards an anti-inflammatory state. So why is that important? It's important to gut health for sure, but the information also goes systemically. So it travels via the systemic circulation and the lymphatic system to the rest of the body. Okay? So it's not limited to just the gut. So what happens in the gut relays to the rest of the body. Okay, so I know we're going to hear more about this later, but microbes can also alter the brain function through immune pathways, neural pathways, as well as endocrine pathways, and through things like serotonin is produced in the gut, can be taken from the gut to the brain, alter brain function. Cytokines produced in the gut go systemic circulation to the brain. Bacterial metabolites that I was talking about can cross and go to the brain. And the brain can be altered through activity of the vagus nerve. And finally, there's also nervous stimulation coming down into the gut. And it's interesting because things like noradrenaline, dopamine, interferon can act on the gut microbes to turn on virulence gene expression and also to enhance cellular growth of subtype of bacteria. So it means you get stressed out, you have noradrenaline, adrenaline happening. Stresses your gut too, and bacteria can read those signals and change their metabolism based on what your body is doing. So just some final notes. A lot of the release of these things is density dependent, or as I said, dependent on other microbes. So if you get a little bit of microbe A, he releases a signal. But when there's a whole lot of these, they grow, then they talk to each other, they release quorum signals, and they say, we got a whole mass of us, and then they release a different signal. And finally, if there's another one present, all these together will see microbe C and say, uh-oh, we got to change our activity because we have another microbe present. And then C will be released. So it really is dependent on who's there, what they're doing, how many are there, to what the overall metabolism signals released are. And diet has a key role. So finally, effects of microbes, they're highly strain specific, not limited to the live organism because the structural component can also get read. And you can get disease due to an absence of a protective signal or an overproduction of, say, a toxic signal. And I will end there. Thank you.